Good morning, everybody. This is a tough act to follow up, and especially with the day being mostly about entrepreneurship and startups, we were wondering how to get this talk in. And the idea came from actually last evening's conversation with an VC manager and entrepreneur. The difference between scientists like some of us is that I would give this talk 5 years later. I'd be like, I don't have enough data. Let me do this some more, and then some more, and then some more, and maybe we'll have a talk in 5 years. Maybe we won't. And the VC fund manager was like, you have some data. Take that risk, show it, let's discuss. Maybe there'll be capital generated, and you can do 20 more things in those 5 years, and not just this one thing over and over again. And that's the idea for today's talk. We're going to present some of our early data, very early data, as part of the Longevity India initiative, which involves understanding how immune cells are changing in individuals as they age. And this is what we have titled as a perspectives from the BHARAT study, which we are conducting. So just to give you a quick overview of what we know about immune cell changes outside India. In India, we don't know as much, at least the Indian population, among the Indian people. This is a very nice sort of overall depiction of what happens in 2015. This was a study done. And basically, what you're looking at is a single infection. Influenza infections across the world have driven some of the early studies on how immune system ages. And what you basically see here is if you have seasonal influenza, and the y-axis here is the number of people that die because of this disease, you will see that its majority is above 70 or below 10. And this is the fundamental idea. Infections kill very young and very old much more than this population that's 10 to about 55, 60. There seems to be a cutoff in the early ages and the old ages. Why is that the case? And there's a bunch of studies that have been done over the last 40, 50 years to show how immune cells change, and this is a summary, and this is not a very good summary. We'll get to some more of the summaries, but basically many of the immune cells are changing, and this is expected, right? So a quick overview of all the changes. If this is all you take, this is a slide you take from it, right? Basically, every immune cell, every organ is changing, just like any other organ system. And this summarizes some of the basic changes in T cells, dendritic cells, macrophages, granulocytes, basically suggesting that some phenotypic markers, these could be used as diagnostics. That is, there are protein level changes on the surface that can be used as diagnostics. And some functional markers or functional effects of the cell are changing. And this is what potentially causes a higher risk for infections or higher chronic inflammatory disease, higher autoimmune disease in older individuals. But what we do not have yet is a clear, there's some association, but there's no clear mechanistic understanding of this change in the immune cell changes this in an aged individual that results in morbidity or mortality. And that's what is missing, and that's what we hope to do as part of the study in IISc, but also that should help us understand how to treat these diseases. So to quickly go over some of the other studies before we get to our studies, so I give you a perspective of what is known. We look at majority of the studies seem to be done in mouse models. Maxim actually was supposed to be here from the Franco-Indian campus. This is a very nice review article from Maxim's group on single cell immune analysis in the mouse. And we now know in the mouse mechanistically what is changing and what contributes to what disease. That same understanding is what we hope to bring in humans, and there are many studies across the world doing that. So to quickly get into some studies that have actually shown specific changes, this particular summary, and it's a very busy slide, and I wanted to show this busy slide, not trying to explain it, but basically this is from the Buck Institute of Aging, showing that there are multiple things that change with age and in obesity, all of which leads to chronic inflammation, which results in B cells that mature in the bone marrow and produce antibodies. They start to change, they become exhausted. So as you age, there's enough evidence now to suggest that B cells become exhausted, and they produce more autoantibodies that will now interact with your own proteins, resulting in inflammatory diseases towards a spectrum of autoimmune diseases. So what's known is that B cells directly contribute aged B cells directly contribute to certain types of autoimmune diseases and inflammatory diseases. Similarly, a large amount of work has been done on T cells. It's a good summary from a bunch of different groups in the world, and this is a summary from an Indian group, but none of this data, and I want to highlight this, none of this data is from an Indian population. This is populations that are not Indian, and that's the key feature here. But basically, again, a large number of things are changing in T cells. Why am I showing you all this without actually getting into any specifics, any details? The idea is to give you a summary of all the different changes that seem to be occurring and tell you, again, not much is known about the Indian population, right? So a quick summary of what is known about all other populations. Myeloid cells, these are cells that are present from birth to death, and they don't necessarily generate memory. They are increased in proportion compared to the memory generating cells, and they have a decreased antibacterial activity. So it's sort of it's a very difficult thing, ironical situation where you have more of these cells, but they're less active. I don't know if they're compensating that by increasing the numbers. The lack of activity is being compensated by increasing numbers, but the basic idea is they're not functioning very well in killing off bacteria, which is one of their major roles. We showed this data. Some antigen experienced cells are more, that is, the numbers increase, but they have less class switching, meaning that they produce less antibodies that are effective, and they may produce more autoantibodies. And T cells, there are reduced numbers of T cells available to produce good memory against a new infection. And there's some changes in CD4-CD8 ratio. This is a very technical term that is involved in cancer development. We won't get into that, or viral diseases. But eventually, there are surface markers that are altered in T cells that make them more suppressive. That is, they're now not able to kill pathogens very effectively, and they can promote tumor growth, which is why you see both of that happen in aged individuals at a much higher proportion. So knowing all this, and knowing that there was very little data from the Indian population, what we decided to do was the following. We decided to first just sample young versus older individuals. Young is in the 18 to 30 age group. Old is in about 60-year age group. 15 numbers in each, very small numbers if you look at it from a scientific perspective and from a statistical perspective, but gives us some idea. We just take a few ml of blood and perform a whole bunch of screening that is supported by this Beckman-Coulter Duraclone kit, which allows us to phenotype. And phenotype is basically understand the protein expression level, doesn't help us understand the function, which is something that we propose to do in the future. This gives us an idea of what immune cells are changing, what numbers, what phenotypes are changing, and this could give us an idea whether the Indian population is behaving similar to the rest of the world, and if there are unique changes that we need to worry about or at least consider when we look at how immune cell can be improved, their function can be improved with age. When we do this, we get this sort of data, and this is the data that you would be learning to analyze if I was taking a class today, and I've skipped a class. I teach from 10 to 11:30 about how to analyze such data. Sorry. The UMAP analysis, 15-color flow cytometry. Basically, it's a clustering analysis, and what I want you to take away from here is you can just look at these as different types of cells. They're not exactly that, but there are different types of cells that are present in young versus different types of cells that are present in old, and they should completely overlap. And you will see that in certain areas, for example, this area, there's supposed to be a cell type, there is a cell type that's much higher expression with the color code here, and the same cell type has much lower expression in the younger population. And that's the general idea here. We can summarize this now using traditional bar plots, and what we show in our data is that among the Indians that we sampled, we see a big change in intermediate monocytes. These monocytes are a type of myeloid cell that are known to be present in higher numbers in the case of infections. They are more activated, they produce more inflammatory cytokines, but they're already more present in older individuals. That is, at the base cell level, in the absence of an infection. So these are healthy or apparently healthy individuals that already have a feature that seems to suggest that they are more inflamed, at least with this particular cell type. Interestingly and contrastingly, the neutrophils, which are another type of myeloid cell type, in fact, they are 60% of your circulating white blood cells, they are more immature. Here there are more inflammatory cells, here there are more immature cells. Immature cells cannot respond to infections as effectively as mature cells, but to compensate for that, they are more inflammatory. So these more inflammatory cells, these more inflammatory cells. And maybe to compensate all that, we have PDL1 expressing neutrophils, and to the scientists in the audience, these are the suppressive neutrophils that are commonly found in tumor microenvironments that promote suppressive microenvironments in the tumor that prevent your immune system from killing tumor cells. So already in older individuals, you're starting to see these changes, and many of these changes have been reported in other populations. So none of this is unique for the Indian population. All we're saying is that there are very big similarities between these changes and what has been reported before. Now we look at B cells, right? B cells, similarly, the plot, UMAP plot, you're starting to see this larger numbers of these particular cell types and absence of this particular cell type that's present here. What do those cells do, right? So quickly, we know that in older individuals, you expect to see class-switched memory cells. That is, your body has been exposed to greater number of pathogens. So there are many more memory cells. Compared to younger individuals, each of you who are older have seen many more infections. So you expect this. This has been reported, and we see it in the Indian population too. Maybe see it much more because the number of types of infections in India seem to be higher, at least has been reported to be higher, viral and bacterial. But what is different is that those same cells, these are the ones that are class switched and are supposed to produce antibodies, they produce less antibodies. That is, even though they are more memory-like, they are there to protect you against older infections that you've had previously. They don't produce as much antibodies. So more numbers of these cells are required, or you may not respond effectively to the same infection again. In a younger individual, you'll respond very well. You're never going to get the same infection as, or not never, but your chances of getting the same infection again are lower. And in older individuals, it seems to be not true. And most importantly, this is something that has been reported in a few papers, but has not been seen in many other populations. We see it in the Indian population. Remember I mentioned the autoreactive, autoantibody producing B cells? That was from the Buck Institute of Aging. We see that phenotype in the Indian population too. There are many more, this particular type, if they're higher in number, they're less autoreactive. They're lower in number, they tend to be more autoreactive. So there are more autoreactive B cells present. What that means is something that we're not sure about, right? We can do the same for T cells, and I'm just going to skip over this slide and show you the actual data. The number of naive T cells, this has been reported in every population, and we see it in the Indian population too. That is, number of cells, new T cells that will respond to new infections are lower and lower in number in the older individuals, right? So our ability to respond to new infections might reduce as we get older. But we have been exposed to many more infections by the time we get older, so our number of memory cells are much higher. So hopefully we can protect ourselves from infections that we've had in the past, but if you have new infections, you're in serious trouble. And this is what we saw in COVID too, a newer infection causing serious problems in older individuals. We see this with influenza, we see this with very many viral infections that are new, right? But most importantly, and this is again something that's not been shown in many populations, but we see it in the Indian population, there are many more anergic, that is cells that are not able to respond to infections. These are the cells that are increased in number, again the PD1, PDL1 axis, that are known to be increased in tumor environments and promote tumor growth. And this might explain why there are more tumors in older individuals. And it's definitely seen in circulation, in just your blood circulation, right? So these are potential opportunities for us to understand what may be changing in older individuals, potential treatment opportunities, but at the same time, we shouldn't just try to decrease them because we do not know how they interact with every other immune cell type. And it's important to understand that immune cells interact with each other. Just removing one cell type will not solve anything. It will just maybe make it much worse, right? So to quickly summarize all our data, it was a pilot study, just 15 individuals per group, very, very small study. So please take every piece of data that I've presented here with a grain of salt, maybe lots of grains of salt. But the basic idea is that phenotype of immune cells change, a large majority of which is similar to what we have seen in other populations, but there seem to be certain changes that are maybe unique or at least different from other populations in T cells, in B cells, and in myeloid cells. So what next? What do we do from all of this? Well, the first thing that we hope to do is to increase the number. And why we increase the number? There are statistical reasons to do this. We know now that we have to get to an N of about 52 or so to get statistical significance across many other groups. I won't get into that, but basically we want to increase the numbers. But most importantly, we want to do this across the age spectrum. And the basic idea here is to understand whether a particular cell type is going to go this way, this way, this way. One of the four different it could be any number of different things. But with age, some feature of an immune cell type is changing. What exactly is changing is what we want to understand across all different immune phenotypes and potentially functions. And that's what I'm going to come to. While phenotype is great, what really matters is the immune cell function, which we have not even started looking at. And it is tough to look at. We could do it by getting the blood and then trying to culture these cells, but they may not respond the same way in culture compared to in native human blood. So that's something that we're working on as to establish the protocols, but we can measure a lot of these. And some of these you can see as diagnostic markers. You would have heard about reactive oxygen species. I need to eat foods that reduce my reactive oxygen species or other features of that. I need to eat foods that prevent inflammation, cytokine secretion, and so on and so forth. These are things that we can check and then see if specific small molecules or specific lifestyle changes affect each of these functions. And that's what we hope to do in the next 3 to 4 years as part of this study. With that, I will stop and thank a lot of the team who has done this work. Amrit and Suramya did most of the work with the help of Gayatri. All of the sampling was done at Ramaya Memorial Hospital, and a big shout out to their team there. This was funded by the Longevity India and Prashant Prakash Family Foundation, and we received support from Beckman Coulter Life Sciences for a lot of the flow cytometry work. With that, I'm happy to take questions. Thank you very much.
Thank you so much, Siddharth. Vishal, can somebody bring the mic, Vishal?
Excellent presentation.
Thank you.
Question. In the immune system, you always see that a person who's fit need not be healthy. So what are the questions that you're going to be asking in your age groups that are going to help differentiate subgroups?
Absolutely. Great question. We have not started doing that, and that's part of the data science analysis that we need to do. Actually, Narendra is just teaching exactly that. We have that data set. We have the BMI, the blood sugar levels, basic blood biochemistry, and a few other features that are changing, and these immune cell types. So now we need to classify whether the immune cell changes are dependent on some of the basic BMI, blood biochemistry, or something else, or are they independent? And the likely answer is that they're not independent, that it is going to be dependent on certain features. But there are going to be anecdotal or unique cases where everything else is, a person is considered fit or apparently healthy, but the immune cell is alone changing. And that might imply certain epigenetic features, which is also being collected now. So we hope to bring in both environment, lifestyle, and genetic features, combine that and see how immune cells are changing.
Superb. There's a question there.
Yes.
Thanks, for that. So your hypothesis of the phenotypic changes makes interesting odds for propensity to manage infections. Most of the inflammation we see with aging is the non-sterile, non-specific, sort of chronic forms. Any early inferences for what that potential application might be through this work?
Great question. I'm going to speculate, and it's mere speculation. From mouse studies that we have done and others have done, even in sterile conditions, inflammation is higher. So it doesn't have to be activated by an infection. So that's sort of well known. That's not the speculation part. Now, we know that sterile conditions are not possible in the human life. So what we see is that in tropical countries with certain bacteria being in larger proportions, there is a tendency towards the immune system as a whole, not individual cells, balancing out the need to eliminate pathogen, because the fundamental role of the immune system, and my speculation here is, that's the part that I'm speculating, they've evolved to eliminate pathogens. And so what happens in that process is that even though they're not very good at eliminating, they produce a large number of inflammatory mediators, the immune cells, and they become overactive or produce larger numbers of themselves, cells, to eliminate the pathogen. In that process, they damage self tissues, non-specifically. So while the pathogen is eliminated, we have greater immunopathology, and that's the key feature here. And we saw this with COVID too. The virus was eliminated from the body most often before an individual died of complications. And the complications were usually immunopathology, that the immune system had started to damage self tissue, or already had damage and the tissue was not able to regenerate fast enough. And that's the feature that we think is happening and that we should control. Long-winded answer to hopefully a straightforward question from you. I hope you got the idea of what we are trying to do.
I know that there are lots of other questions for Siddharth, but in the interest of time, perhaps we'll move on. And I'm sure he'll be around.
Yes, I'm
Pick
here
off
for
some
that.
more lectures from your circuit, and there'll be questions to handle later on during tea. Thanks, Siddharth. Thanks a lot.